So at that point, when an apurinic site is recognized, there's another protein that comes in and cleaves out the sugar phosphate that's that is there, but no longer attached to a DNA base. And now that hole will be recognized by the DNA polymerase, which will come in and do the patch repair, and then DNA ligase will seal the sugar phosphate backbone so everything's back to normal. Okay. So in this case, it's fairly easy. You know that the damaged base is the one that was taken out, and it just used the other strand as the complementary sequence to then say which it should put there.
Are we going to know like DNA glycolysis or just know the general
process?
Okay.
Yeah, so again, this falls under the category of what happens when the damage is done to DNA and how do we keep the DNA the same sequence so we don't have mutations that come in. We get genetics, we'll go into much more detail with these repair pathways, but I want you to at least see some examples of how this stuff works. Okay. All right, so this is the one that ends up being the most important as far as the repair pathway goes, and we'll talk about a little bit why that's the case. This is what's known as homologous recombination. There's a host of complex proteins that exist here that all work, but we're not going to talk about what they are, but I want you to understand the process of what happened here again. Okay, so this process is used for double-strand breaks or for when replication stalls. So we talked about the beta clamp is used to help make sure that the polymerase doesn't fall off. If the polymerase falls off, you basically will have a scenario where you have an empty strand with the other template strand down here, and homologous recombination can help restart that process as well if that replication falls apart. Okay, so here's what occurs. So if you start with a double strand break, this is easiest to see. At this point, the question becomes, if that break occurred, you don't know, once you have two ends, you don't know where those ends are supposed to be, or if there's a piece in the middle that has been lost, right? So basically what you need to do is, especially if you have two ends or three ends, you don't know which ends are supposed to go where, right? Because at this point, you've lost continuity between the strand. So the way that the cell solves this problem is that there is another copy of the genome in the cell, right? So this is, if this is a diploid cell, you always have a backup. So if this is chromosome 1 and there's a break there, we also have an intact chromosome 1 down here. So that intact one is going to be your key to the question of what happened with this guy. Okay? So if you lost bases in between or if everything's good, this is going to tell you the answer for that. Okay? So the way that this works is the following, is that you have this double strand break. The first thing you do is you need to create a free 3' end. Okay? So in order for that to happen, what happens is that there is a protein that chews up the bases to kind of create that overhang. All right, so it chews both here, chews those bases apart and these bases apart. And a free 3' end is then mediated to do invasion to figure out where its complementary strand is. Right? So now this strand here is finding out where its complementary sequence is on the other intact chromosome. Okay? Right? Because this strand will base pair with one of these strands, and it will be the exact same copy as the other one. With me so far? Okay, so once it has figured that, it can extend that 3' end all the way down, just by the same process that we already are aware of. And then once it does that, it can, once it goes far enough, it should be able to base pair with this guy. Okay, so once that occurs, now you can have base pairing here and then fill in these patches. Okay? So it's using this red strand as a template to figure out what is lost, if anything, and allowing it to then to rejoin and base pair with the damaged double strand break. Okay? So this is used all the time, and this is probably the default error repair pathway for cells, is to use the homologous recombination to kind of direct the repair. All right, so why don't we explain this one to your neighbors, and then I'll entertain questions before we talk about telomeres.
And then like, why is this and then passing it just
Yeah, so the equation minus the complementary function, and then
to me, it's
and then one of the this is or it does not feel it's so like perhaps these just go straight
until you get to the
right, but if that's the case, it still doesn't know if these two entities join together or if there is a thing that is helped to figure out. But then we'll go right into this one. And if that's the case, then that's the number that you say we should go in here. Yeah, this previous representation. So it has these bases with these exact complements, right? And they got each way here, right? So it may or may not, but either way, this process will make sure that they get sealed in the right balance. It will, and it's helping open that strand up so that it actually can scan and figure out where the complementary sequence is. Then once that happens, at that point, we have a situation where now the strand can be extended with DNA polymerase.
So it's just a protein that's responsible for figuring out where it should like invade.
Yep, yep. Oh, both help it invade as well as help scan to wait to see where the complementarity exists. And you can imagine that's the same thing. They're looking for exact base pairing, because if it's not exact, you're going to have these bubbles, and that protein's going to look for that same sort of scenario where you have this exact perfect base pairing for that sequence with what it's scanning against. Okay, other questions? Yeah?
What is a double strand break then? Is that just like a mismatch in both of the strands?
No, it's a physical break of the chromosome, right? Where it's literally snapped in half, right? UV radiation, oxidation events sometimes will do this. Physical contortion, we talked about where if the DNA is twisted too much, that it actually could cause it, right? So any of these sort of things could have the DNA actually cause to be snapped. So this is a problem in the sense that if the DNA, if it can't do this, and it finds two ends, this is a freak out for the cell. Like this is all red alarm fire. Like you do not find ends of DNA in the cell, like never. And so ends of DNA, the cell will start putting, the other backup to this is what they call non-homologous end joining, and that's basically taking two ends and just putting them together and hoping for the best. So this is genetic diseases result from that. So if you have a break and you take chromosome 6 and you put it with chromosome 4, you can imagine that sometimes that works out well for you, but most times it does not, right? So this is our first plan. If we can do homologous recombination, we can repair that double strand break. And if we don't, then the cell just puts ends together and hopes. This will work. All right, let's go on. And then just sort of put it in the corner and just walk away, right? Oh no, who did that? Right. Okay, ready? Good? Questions? All right, so that leads us to the, so this, I'm going to skip, this is basically the steps, right? RNA primer, you make it, you do it. This is reviewing what we've already gone through several times. And I could show you that the same sort of video that we showed before, but we're going to skip that too. I'm going to go to telomeres. Okay, so I just said that ends are problems, that you don't see ends. And that's actually true. So in bacteria, what they do is they have a circular chromosome. We don't have a circular chromosome. We actually have ends. So, and I just said ends are freakouts. So for telomeres, our ends need to be covered. They need to be hidden. And so I'm going to show you what that looks like. So the first thing is that these telomeres, you can see that they don't contain any genes. There's not genetic information there that the cell's going to use. They tend to have, there seems to be some connection with the size of your telomere to the sort of the clock, your how old, your rings on a tree. This is similar. You look at your telomeres, and it seems to be able to tell how many divisions the cell has gone through. So you can see that embryonic stem cells, about 20 kbs of telomere repeat, and there is literally just repeat sequences one after another after another. And if you can go down to somatic cells, every time that you go through a replication event, you're going to cut off a part of your telomere. And we'll talk about why that's the case, because it comes down to the lagging strand. I'll show you how that ends up happening. So, so a lot of people thought this was the key to sort of eternal youth, is that if we could just fix the telomeres, then that would give us eternal mortality sort of thing. And that ends up not being true either, because cancer cells have actually have processes in place where they actually extend the telomeres, and that seems to make the cells not just immoral, but uncontrolled regulation, where they just grow and grow and grow. So there seems to be, again, a Goldilocks scenario here where that there's a good, happy medium, and if you go too short, the cell actually will then move itself into senescence or actually into programmed cell death. Whereas when the telomeres get so short that now the genetic information that the telomeres were protecting, the cell recognizes that and says, we're done. We don't have enough telomeres left before this genetic information is going to start to be compromised. And if that's the case, we just need to fall on sword and go on with having other cells replace us. Okay? So these telomeres act as that sort of sensor, but you can see that they also have consequences for in cancer, that you just can't have them, infinitely long. That also has consequences. Okay? All right. So here's what the telomeres look like. So the problem with, so you can see the first thing I'll say is that I said ends are a problem. So telomeres end up causing this looped structure because we don't want to just have an end of DNA in the middle of the cell, because the cell will try to join it with whatever it finds, or it will try to repair it. So it forms these T-loops. Basically the whole thing integrates into make a triple strand and makes sort of just like a little circle structure so that the cell doesn't see an end and gets scared with that. Okay, so that's the first thing. But the second thing I want you to get is this part here, is that you can imagine that when you go through replication, so here's, right, leading strand synthesis. So here's our end, right? So our fork, right? So here's your leading strand synthesis. It has no problem going right to the end of the chromosome, right? The problem is on the lagging strand, that's not as easy, right? Is that lagging strand is going to be here, and it's going to do a fragment this way, and another fragment this way, and another this way. Unless that you are able to position this RNA primer at the very last base, like right here, and have it prime back, you're going to lose some part of it. Does that make sense? Right. So this is the problem with telomeres, and this is why telomeres exist. Is that this is like asking the RNA primase to jump onto the very edge of a desk and hit right here, and then prime back this way, as opposed to the leading strand, since it just goes until it falls off. Okay. So that's a really tough ask of a protein to be able to land exactly on the end. So it will sometimes land here, sometimes here, and sometimes way over here and just fall off, right? So whatever the case may be, is not likely going to get to the very end. And so whatever portion of a lagging strand, this is what you're going to lose, right, out of your end of your DNA. Okay, so that's the end problem, is that the lagging strand synthesis can't land exactly on the very end and prime backwards. So what ends up happening, and this is only true in embryonic cells or in cancer cells, they have an enzyme called telomerase, and telomerase actually can extend the length of the telomeres. Okay? In all the other cells, what happens is what I just showed you. The telomere gets short. Every generation it gets shorter by whatever that lagging strand synthesis wasn't able to prime. Okay? Notice that what telomerase does is it actually has an enzyme, and it actually in the enzyme itself has a template for the telomere repeat. And so it can actually extend the length of the telomere by using this as a template, and then having this enzyme actually fill in those blanks. Okay, so so in this way, this is how telomeres can actually expand out, is by having this telomerase just keep on using this template to extend out the repeat. So in cancer cells, they actually upregulate these telomerase enzymes to actually extend out the length of the telomeres, and you can see that actually causes some significant problems where they just grow unchecked for this and for other reasons. Okay? All right, so I'll give you a chance to process. So explain to your neighbor why that in normal somatic cells that telomeres end up shortening, and relate that to the lagging strands, and then tell me about how the telomerase enzyme works to combat that. All right, go. Stem cells, or stem cells in general, and in cancer cells. The rest of our cells lack that enzyme, and therefore our telomeres are growing shorter and shorter.
So the older the cell, the shorter the telomere?
Yes. Yep. Was that question, Alicia? Good.
Well, because like as you age, then you eventually like run out of telomere that can like speed up. So is that essentially when your cells just like slow down like replication?
Yes.
And you start to see like wrinkles.
Yes.
And stuff like that.
Yep.
That sucks.
Yeah.
20.
Right. So this is why the as you age, that everything begins to slow down. A lot of it has to do with telomeres and how long they are. I mean, there's other things going on there. There's signaling events that have to do with your thyroid hormones and other things, but this is a piece of that. Yep.
Question for you. You might not know the answer to this, but like I don't know if you keep up with like makeup or like fashion. So like people have
All the time.
Yeah, I know, right? I think so. So people get those new like facials where they will take like your blood and they'll like basically spin it up really fast, and then they'll like insert parts of it back into your face to like prevent wrinkles. Does that have to do with like stem cells or anything like that?
It's a good question. You know what? We have this assignment on Friday about reading the literature on some subject that's interesting. You should check it out. I don't know, but I know that there's literature out there on it. So
Okay.
why don't you find it and then post it to our discussion board?
So I'm just gonna read about facials.
Yeah, please do, because I'm in dire need of one. And I want to make sure if I get one, it's the best. All right. Any last questions on telomeres? All right. So let's switch to transcription. All right. So this whole first unit is around DNA. So far that what we've done is covered the process of, well, the DNA structure itself, and now we've gone through replication, and now we're going through transcription. And then we'll talk about transcription here for a while, of controlled transcription, and then we'll talk about translation. All right, so we're basically taking DNA and following it all the way through from DNA all the way to protein. Okay, so transcription, which you already are familiar with from our reading of the paper, is the process of going from DNA into messenger RNA, right? So this is the intermediate step before we get to proteins. Okay, so we are interested in understanding how this occurs as well as how it's controlled. That's our kind of goal. Okay. Here's my goals for what you should be able to do at the end of this chapter to kind of focus our efforts of where we're headed. Okay, so here's the first starting point. So when we first understood DNA, the first hypothesis was that one gene is related to one enzyme. And then as we understood more, we realized that perhaps that's not exactly accurate. Then we said, all right, then one gene was equal to one protein. And then we found that that wasn't quite accurate. So then we said one gene is one polypeptide, and we turned out that wasn't all that accurate. And then we said one gene equals several polypeptides. So our current understanding is that last one, that we understand that one gene could produce several different polypeptides. And you should make an asterisk that this is true mostly for eukaryote organisms. And it's because of this alternate splicing. Are you familiar with alternate splicing, what that means? Okay, so let me run through that. So you are familiar that genes have exons and introns, right? In eukaryotes. This is not true for prokaryotes. But in eukaryotes, there are exons, which are the final versions, or the parts that are actually going to be spliced together in order to make the final protein. Okay? So the mRNA, what needs to happen is that we will produce this, but we need to then have these exons be put together to make the protein sequence that's going to be actually lived out. Okay, so you can see that this mRNA, after it's been processed, produces this introns 1, 2, 3. Right? So the process of transcription will take the DNA and will make an mRNA transcript that contains both introns and exons. That mRNA needs to be processed, and we'll talk about this in this chapter. That processing does a couple different things, but one of the critical things is it splices out these introns and puts together these exons. Okay, so you can imagine that if you have 1, 2, 3, 4, there's a couple different ways you can make to mash your splicing, and therefore you could make several different proteins from that. You can make protein A, which is exons 1 through 3, and you can make proteins B, which is 1, 2, 4. Okay? So depending on how you process the mRNA, you can produce several different polypeptides. Okay? So in prokaryotes, for the most part, this is true. This does not exist. Prokaryotes, 1 gene equals 1 polypeptide. That ends up being true. Okay? And the reason why we say that if 1 gene doesn't equal 1 protein is that proteins oftentimes are made up of several different polypeptides. So meaning that there will be several different genes that come together to make 1 protein. So that's why that second bullet ends up being false in some cases. Okay? Questions for me on that? Of where we're at in that understanding? So this shows you the scope of where we're headed. We're going to be talking about the process of transcription, which shows you this, but then also once you have the transcript, how do you process it into the final form of the mRNA? Because once we're at this point, now that we have an mRNA, then we can go to translation, which is where we're headed after this chapter. Okay? So this gives you a run through of where we're headed. How do you control this process of transcription, and how do you process that final transcript so it's ready for translation? Okay, so what does the process of transcription? This process, remember, transcription is going from DNA to RNA. So if you're going to make an RNA transcript, you need an RNA polymerase. So you use a DNA-dependent RNA polymerase. Basically, what that means is that you have a DNA template that is used to then make RNA, and that RNA polymerase lays down those RNA bases. Okay. So the promoter. You've heard that language before from our paper. The promoter is the site where that polymerase is going to bind and where transcription is going to start. So, and there's lots of different factors that help the RNA polymerase bind to DNA, and you're familiar also from our paper as well. There's activators, there's repressors, and we'll talk about that, but there's things that help stabilize the polymerase at the promoter to help encourage it to start transcription. Okay? Direction of transcription, always the same as replication, 5' to 3'. Right. So this RNA polymerase described, do you think that it could be, you do the RNA priming work required for replication? Turn to your neighbors, see what you think. Go. And we said that the DNA polymerase couldn't do that, right? It needed what did it need? So when you look at the DNA polymerase to start replication, what does it need? Start what? Oh, so careful of your language. That's not right for lots of different reasons. So start codon is what?
Translation.
Yeah. So that's a triplet codon found in. Start codons are found in what? Where? In where?
The mRNA.
The mRNA, right? So start codons are start codons for translation. So what do you need for DNA replication? Not a start codon. What is it called? Yeah, the primase is the enzyme, but what's the sequence called?
Promoter.
Nope. Promoter is found where? See this. So this is exactly what you need to think about, right? Is that when I get to an exam, I'm going to go to these same places because I know that they're confusing, right? And language is really critical, right? So a promoter is what? Is not translation. Transcription, and it's found where? Yeah, see, now you're seeing where the fuzziness is going to come.
Will you say that again?
Where do you find a promoter? In what? Do you find it in a protein? Do you find it in DNA? Do you find it in RNA? And what binds it? Yes, you do. You read a whole paper about it.
Whole.
Yeah. Which one? So,
Two.
all two, right? Yeah. So promoters are found. Yeah, transcription start site. But what is that? Is that a piece of DNA or a piece of RNA?
DNA.
That's a piece of DNA, right? Promoters are places where transcription begins. So you should be able to find them in the DNA. Okay? What is the sequence that starts DNA replication?
An activator.
Mm-mm. That's transcription too.
Someone else did it.
Yeah.
RNA
You're,
primer.
huh?
The RNA primer.
Yeah, it will start at the RNA primer, but how did you know that to start there? It's called an origin. Remember? Yeah. The
Oh,
origin.
Way too hard.
Yeah, see, the origin is the site where replication will begin, and there are several different origins that will be across the DNA sequence, right? So what happens is that at the origin, you're gonna remember that's where you load the DNA A and the helicase, and then the polymerases come in. Remember we talked about that last time? That's what we're talking about. So this question, back to here. RNA polymerase 2 is the primary mode of what's doing transcription. It binds to and recognizes what type of sequence? If you're going to start transcription, where does the RNA polymerase come bind? The promoter, okay? This is practice words, right? So RNA polymerase will bind the promoter to start transcription, okay? If the RNA polymerase will bind to the promoter, that means it's not going to recognize an origin. So there will be another RNA polymerase that will bind to an origin type sequence and start the process of putting down an RNA primer for replication. Yep. So,
I need like a recording.
yeah, right? Talk to Dom. So, so you need to practice these words because I know that they all blur together, but it's really important that you be able to use the right words in the right sequence, because otherwise I can't tell if you understand it or if you just misspoke. Both are likely, right? So practice your words. So let me ask another question so you can practice. Do you tell your neighbor so there's DNA polymerases and RNA polymerases. Tell me where they bind, what they do, and what type of nucleic acid they bind in that process. Okay? So go through the process of DNA polymerase. What does it bind to start its activity? What's the process it does? And what type of nucleic acid does it bind to and produce? Okay? Lined up in the right boxes. Okay, so origins are sequences of DNA at which replication process begins and is initiated. Okay, so promoters are sequences of DNA at which transcription, the process of transcription, begins. Okay, so notice that these are just kind of landmarks or flags to say this is in the DNA where we're going to begin the process of replication. This is in the process. This is the position in the DNA at which we're going to begin the process of transcription. Right. So when we talk about those things, those are literal sequences in the DNA, but they're important because those are the sites where different proteins will bind to begin those processes. Right. So we know that there are certain proteins that will bind the origin to begin the unwinding and then the process of loading on the replication machinery and starting the process of replication. At other points in the cell cycle, during growth, we want to make genes. We want to have proteins being made, so we have to go through transcription, which is where promoters come in, and transcription start sites, and activator elements, and all those other language terms that come in there. Okay? So just kind of, I think the first thing that you need to do when you go down this route is, what processes am I talking about, right? Get yourself zoned into saying, all right, I'm talking about transcription now. So all the other terms that have to do with translation and replication need to go outside my mind because they're not relevant at this point. Okay? What's still fuzzy? We'll keep on going. So we're just starting transcription. So I dove probably deeper than I should have because we've covered a lot of this in the paper. But now it's make sure that you have the paper content in this section of the course, right? Do not try to apply anything from the paper from what we just talked about with replication, because that's not what the paper was about. The process, the histone phasing and all that stuff is about transcription, which we're talking about right now. Okay? So one other thing, transcription start site versus promoter. They're not the same. We'll talk more about this, but promoter is the site of binding for the RNA polymerase. This transcription start site is exactly what it sounds like. It's the first base of transcription. Right? There will be one base that is the first one in the mRNA chain. That's it. The transcription start site is base 1 of the mRNA transcript. So anything before that base will not be transcribed. Okay? We'll get to that. That's the difference in those terms. Okay? Okay. So let's talk about transcription. Okay, so I'll circle back to talk about how it initiates, but here's what it looks like, writ large. Okay, so notice that what you see is here's the RNA polymerase, and it has initiated to form this bubble, similar to what you saw in the DNA replication process, right? You have to form a bubble to kind of bring the two strands apart from one another. And then what happens is that you begin to form an RNA-DNA hybrid here, and then you're just going to wind and unwind the DNAs as you travel through it. Okay? So this process is producing this mRNA strand that comes out here, and at some point at the very end here, we're going to stop the process of transcription. So we'll go through this, but we're going to talk about how do you initiate it, how do you transcribe when you're doing it, and then how do you end it, okay? And the different terms that go along with that. Okay? So this is what it looks like. We're going to dive to the details of how this happens. Questions on this? Still going 5' to 3'. Notice that you're creating 5' to 3'. This is your 3', and you just keep on moving yourself down in that direction. Okay? All right. So let's keep on moving. So so I want to talk about, we're going to start with just prokaryotes because it's a lot simpler. And then I'll briefly show you eukaryotes and overwhelm you, and then I'll very quickly move away from that so that you don't get too nervous. Okay? So let's start with prokaryotes. So RNA polymerase is just not one enzyme. It's made up of 5 different massive proteins. This is a huge machinery. So that's the first thing you need to get. I'm not interested in you knowing all of them, but you do need to recognize a couple. So the first thing you need to know is this guy. So the sigma factor is one of those 5 of RNA polymerase. And that sigma factor, its job is to recognize the promoter sequence. Okay? That's all that the sigma factor will do. And you can see is once you get downstream, 10 to 12 nucleotides, the sigma factor will release. And the rest of the polymerase components will keep on going. Okay, so this is important to think about, right? Is that if the sigma factor job is to find the polymerase, its job is to recognize and bind promoters. Okay, so basically all it wants to do is bind to that sequence. Okay, so as the polymerase begins to try to move down the gene, the sigma factor is still trying to hold on to the promoter. This is why it releases at 10 to 12, because eventually the force of the polymerase wanting to go and transcribe and the force of the sigma factor wanting to stay by the promoter is too much, and they just kind of split. Okay, so that's the rationale of why the sigma factor lets go, because it never really wanted to do transcription to begin with. It wants to bind promoters. Okay, so it will be tried to be drug along, and eventually the polymerase will escape. The sigma factor will just remain to where it is. Okay.
So then does the sigma factor go and find a different one?
Yep, it will either start, so all that sigma factors do is bind promoters and then recruit polymerases to come. So it will bind another promoter and recruit another polymerase, or it may just start another transcript right back to where it was. Like it will bind to that same promoter and another polymerase will come in and it will start transcription again. So you have 2 different copies of the mRNA coming off. Okay. So that's the sigma factor. So the promoters, the thing here is that there has to be a sequence, a specific sequence that the sigma factors are looking for. So that's it's called a consensus sequence. You basically are looking for certain combinations of ATs, Gs, and Cs. And this, we'll talk about the print mailbox as well. I'll show you that here. Okay, so I'll come back to Rho. We'll just pause on that for now. So I'll show you the promoter and then we'll stop there. Okay, so the promoter is 2 different sets of sequences, the -35 and the -10. These 2 things together comprise the promoter. This is what it's looking for. The sigma factor is looking for when it's trying to find promoters. And that's the consensus sequence. I don't want you to memorize it, but you can see that it's looking for TTGACA and TATAAT. That by definition, if you say what's a promoter, this is your technical definition. That's these sequences. And what they're saying is the start point, this is the transcription start site, the +1. So the promoter is 10 bases upstream of that and 35 bases upstream of that. Okay, that's what it's looking for. All right, I think we're done, but we'll start here Wednesday.